Good afternoon, everyone. We are live. Welcome to Streaming Strawberries. We're here in the Gulf Coast Research and Education Center in Waimama, Florida. Remember that. We are here with four amazing scientists at the research, and we're gonna introduce them one by one. But first, my name is Cesar Zamora. I'm with the Streaming Strawberry and the Streaming Science team, and we'll introduce everybody else in a minute. Okay. First, let's meet the scientist, and we'll start to my left.
Hi everyone, I'm Natalia Peres. I'm a professor here at the University of Florida Gulf Coast Research and Education Center, and I'd like to welcome you all to our lab.
Hi, my name is Teresa Sejo, and I am a senior biologist. I'm a researcher working for Dr. Natalia Peres, and I work both as a researcher and as a diagnostician in our plant disease clinic.
Hi, hello everyone. My name is Marcos Marin. I am assistant scientist here in the GCREC Center. I work for Dr. Natalia Peres and coordinate the Plant Diagnostics Clinic.
Hello everyone, my name is Andrea Marquez and I'm a lab technician, and I'm also working here for Dr. Natalia Peres.
Thank you very much, guys, for introducing yourself. I'm very excited to go on this journey and talking with you all about what you do here and learn more about strawberries and how we grow them in Florida. But first, let's introduce our Streaming Strawberries team, our Streaming Science team. On the director, we have Shannon. Say hi, Shannon. Camera 1, we have Jessica. Jessica, say hi. Camera 2, the one that you're looking at right now, we have Bethany. That's Bethany. And our Q&A, we have Caitlin. Please keep Caitlin busy and send your questions. We really want to see them and have the scientists respond them, okay? Alright, so we are here at like I said, we are here at the Gulf Coast Research and Education Center. This is a facility. We're right now in the Plant Diagnostics Lab. It's a facility that grows and studies strawberries and strawberry diseases, and we're going to talk a little bit about that with Dr. Peres, okay? Dr. Peres, what do we do here first?
Okay, so well, let me start where we are, because I hear there's people that are not even from Florida here. So we're based on a research center that's just south of Tampa, and our county is where the majority of strawberries are grown in Florida. We grow 95% of our strawberries here in this county. So a lot of the research we do here is research to help those growers to grow better and healthier strawberries.
Yeah, okay, so that's why the place is called the Gulf Coast Research and Education Center, because we're in the Gulf Coast near Tampa.
Yes, that's correct.
Okay, let's get to the questions. So, we're here. You guys study here plants, strawberries in specific, and strawberry diseases. So I hear that strawberries get sick, and so if you can talk to me a little bit about that and why is it important to know that strawberries get sick.
Okay. Yeah, well, not only strawberries, but all plants get sick, just like we do. I mean, the reason we work with strawberries here is because, like I said, this it's very important for this county economically. It generates about $300 million a year for the county. So it's a very important crop. And so the research we do is trying to help those growers whenever a plant gets sick. We have to figure out what it is and what's causing, and sometimes it becomes like a whole research project for us, trying to come up with better ways to control those diseases so they don't have huge losses in their fields, basically. So we, and just a little background on how we grow strawberries in Florida, because that's different if there's people connecting from up north. Strawberries can be grown as a perennial crop, like for more than 1 year. In Florida, we grow strawberries as an annual crop, which means we plant them every year. We start with land preparation about in August, September. We prepare the beds, we treat the soil, we put the irrigation, and we were just planting them. We're not in the field right now, but we just planted them a few weeks ago. We use transplants. They're not transplanted from seeds. Some people might think they're planted from seeds, but we actually use baby plants. They're like multiplied in nurseries. They're miles from where we are now. We get those transplants, and they're planted in the field one by one. And when they come to Florida, we often think, or they look like healthy, but once we plant them here, because of our conditions, it's very humid and warm. As you know, it's 85 degrees today. Then some diseases might start showing up, and those diseases will manifest in different forms. It could be leaf spots, it could be crown rots, it could be affecting the roots, and you're going to learn about this in the next segments. I don't want to steal their thunder there.
Yeah. So you're saying that in Florida you get all these diseases. Is it like you said, it gets warm in Florida? So is it the weather? Is it one of the states that gets the most diseases in plants, or only in strawberries? And also you said that they plant them annually, so meaning just once a year, and that's all we get in the supermarket. And so how else do we get strawberries in the supermarket? Because I see them right now also.
Okay, so for that second question, so we only plant this part of the year. Florida is known as the winter capital for strawberries. It's about the only state, the only area that can grow strawberries during the winter. And that's why it's very important economically. There's not a lot of competition at that time until fruit starts coming from Mexico or other areas. The reason we have strawberries and all year long in the supermarkets, because they'll be coming from different regions depending on the time of the year. But during the winter, the majority of what you find in the market is going to be hopefully from Florida. The first part of your question, I don't remember.
Yeah, no, the first part of my question was, what makes Florida so prone to get diseases, or for strawberries to get diseases?
Okay, yeah, it's like you said, the warm and humid weather. I usually joke as a job security for plant pathologists like me.
Yeah. So, and a plant pathology is someone like you who studies diseases in plants. So that's what pathology means, or a pathologist.
Yep, exactly. Plant pathologist. So we study diseases in plants, not only in strawberries. So my degree, I study citrus diseases in citrus. So we study diseases in all different crops, and there are specializations within plant pathology. Most of the diseases we get on strawberries are caused by fungi, like the fungal diseases, but there are diseases caused by other organisms like bacteria or virus, and within plant pathology there are specializations as well, as like plant virologist or bacteriologist, or in my case I study more the fungal diseases, because that's what we get the most on strawberries.
So it sounds like you guys are like any other doctor that goes into studying all this and then just specialize in something specific. I
Exactly.
really like talking with you about this. I can't wait to hear. I can wait to hear more about what the center does. Thank you very much for being here with us, and thank you guys for connecting as well. And now we're gonna go to our live Q&A. So keep sending your questions. We're gonna go to Caitlin, and she's gonna ask questions to Dr. Peres.
Thank you so much, guys. We have had a couple of questions come in already, so don't forget to submit your questions, and Dr. Perez will answer them. We also are going to be pushing out some polls, so we're going to be asking you guys some questions while you're watching, so you guys can feel free to answer those polls for us, so we can get a little bit of information about where you guys are at, what you're doing, and what you're learning today. So our first question is from Kylie, and Kylie wants to know, Dr. Perez, what is your favorite part about studying strawberries?
Hi, Kylie. Thank you for your question. Thank you for joining. I think the best part for us here, not just for me, is that we do research on strawberries, and I think in the next segment, please make sure to join the next one. We'll be showing the field is that we do the research, and then we get to harvest, and then at the end we can take those berries home and enjoy them.
That's awesome. The next question is, how do you get strawberries to be sweeter?
That's a very good question. It's mostly through breeding, and we're not going to talk much today specifically about breeding, but there is a different program, a different group of researchers here that basically what you do is that you make crosses and you try to select the cultivars that are sweeter. That's one big aspect for sweet strawberries. Like there are types of strawberries that they were bred, just like you breed dogs, you breed strawberries to be sweeter. That's one big component. Another big component is the weather. So for us in Florida, we usually get the best strawberries when the nights are cool and the days are sunny. That's when they taste the best. So even like a cultivar that is bred to be sweet, that flavor varies through the season. So it can be sweet one week and the next week not as sweet.
This isn't a question that was asked, but it is a question that I have from what you just answered. So I know that you guys will be showing us hopefully the field the next time that we go live. Is there any research that you guys do here about making strawberries sweeter out in the field?
Yes, there is actually a big component of our breeding program. We work very close with them. The main aspect of their program is to find strawberries that are sweeter, and then we work with them to make sure those strawberries are not only sweet, but they also can resist to all those different diseases that we have.
That's awesome. Thank you so much. And we have one more question, or two more questions. One is from Steph. And Steph wants to know, did you always know that you wanted to be a plant pathologist?
Oh, hi, Steph. That's a very good question, and the answer is no. It's something I found along the line. I study agricultural sciences, which was very broad, and during that time I started internship with plant pathology and really enjoyed that, and then ended up doing a master's and then going for a PhD, and this is what I love now, but it's not something I always liked biology. In school and middle school and high school, I always liked biology, and I wanted to do something apply where I could apply biology, and this is a really good career for that applied biology that I was looking for.
That's awesome. And the last question for this Q&A segment before we throw it back to you guys is, what is the biggest issue with strawberries in Florida?
Oh man, that's a very big question. I can say it's diseases because that's what I work, and there are some diseases that are important. But thinking about the industry in general, their major struggle right now is cost of production, and especially labor. It's very expensive to grow an acre of strawberries, and that labor to harvest the strawberries, it's all manual harvest, and it's very difficult to hire people that want to do that job. So we often have to bring migrants to do the work and offer housing and everything for them. So it's very, very expensive. So for the growers, I would say that's their number one challenge. And then, of course, there's pests and diseases that can, even after all that investment, can cause losses to that investment. So that's what we are here for.
Thank you so much. And guys, don't forget to keep submitting your questions. We're going to bring it back to Cesar.
Thank you very much, Caitlin, and thank you all of you to send questions. Keep bringing them in. We all love answering your questions here, so just keep us busy, please. And thank you, Dr. Peres, to be here with us and to tell us what the Plant Diagnostics does. And we want to talk to more scientists here and learn more about the place.
Yeah, thank you, and thank you you all for joining.
All right, guys. Now let's get back to to topic here. So, as a recap, we're here in the Gulf Coast Research and Education Center in Waimama, Florida, in the Gulf Coast of Florida, which is near Tampa, ergo the name. And we have learned about what the Plant Diagnostics Lab do. And like you heard, there's also a field component here because to study strawberries, you need to grow strawberries. So if you want to know about that, please tune in on November 16, and we're going to be outside in the field working with that. But without much further ado, let's get to the topic. Now, let's go talk to Teresa about how the scientific process work here. Hey, Teresa.
Hi. So when the farmers observe symptoms in the field, their plants are not growing well, they may be dying, they may be smaller than they expected to be, they may have spots on the leaf, they may start turning a funny color and look yellow instead of green. They're going to want to know what's wrong with their plants. And so they're going to bring us plants and say, what is wrong with our plants? And the first thing we're going to do is ask them to fill out a form and tell us about what their plants. We want to know what cultivar of strawberry they're growing. That would be like a breed of a dog. So what type of strawberry they're growing. How long ago did they plant it in the field? What symptoms are they seeing in the field? What percentage of their plants are showing these symptoms? How long have they seen the symptoms for? Are the symptoms located just in one part of the field, like a part where it's really low and they might get flooding, or are they all over the field, spread evenly, or are they occurring in random patches? And when we get this information about what they're seeing, it helps us begin to diagnose the samples. The next thing we're going to do is actually look at the plants. So right here, I actually have some strawberries that were from our strawberry field that I pulled this morning. And I have some nice healthy strawberries that are doing well. These were planted about 2 weeks ago, and they look beautiful. And then I have plants that were dying. They weren't doing well. These did not establish. So these were planted at the same time. They were in the same field. They were right next to each other. And they were the same size when they went in the ground, and you can see the difference in the size. This is just not growing at all. I don't know if you can see the roots here. The healthy one has some lighter colored light brown roots where there's no new root growth on this plant. On the one that's having problems, the roots are black. And when the plants start dying, we often suspect that a part of the crown is infected. So I'm going to cut open these plants, and so the crown of the plant is this part at the base of the plant. It's like the bottom of the stem. The leaves come out of it on the top, and the roots come out in the bottom. It's the part that's right at the top of the soil. And if I cut open a healthy plant, it's nice and beautiful and white inside. If I cut open one of these plants that are small and wilting and did not grow, have black roots, look at that crown. It's very red, only got a little bit of white at the very top. That is crown. This plant is rotting from the crown of the plant. So now I have an idea of what we're looking at. We're not looking at a leaf spot, we're looking at a crown rot that's killing this plant. But that still doesn't tell me what it is, because there's a lot of different diseases that can, a lot of different things that can cause crown rots. Okay. So plant diseases can be caused by either abiotic or biotic. So sometimes the symptoms that growers observe are not actually a disease. They can be caused by things like too much fertilizer can cause a burn, and the edges of the leaf may burn, or if you don't have enough fertilizer, your plants may start turning yellow. If they don't get enough water, they can wilt from drought. So sometimes it's not actually caused by a pathogen, but a lot of times what we're looking at in here is diseases that are caused by fungi, bacteria, viruses, sometimes little tiny microscopic worms called nematodes, and also insects. And all of these can attack plants and cause diseases.
Yeah, you said something that maybe the kids want to learn a little more about. Biotic and abiotic. What does that mean? What's the difference between the two?
Okay, so abiotic is something that's is a problem that's being caused not by a living thing, by fertilizer, by herbicide, by the weather, by lack of water, by too much water. And a biotic problem is something that's being caused by a living organism, such as an insect or a nematode or fungi or bacteria.
Okay, so the biotic is pretty much what you guys study here the most, and you also said that you have some samples for us to see in the microscope. But
Yes.
if you want to talk a little more about that, and then we can just move to the microscope, that would be fun.
Yeah, so in our lab we focus mostly on fungal diseases and a little bit on bacterial diseases. Strawberries have a lot of different fungal diseases. They only have 2 bacterial diseases in their leaf spots. That's not true of other plants. In other plants there might be many different bacterial diseases. We don't have a huge problem with viruses in strawberries, so we don't have to do a lot of work with viruses. And if we get insects or nematode issues, we actually have a nematologist and an entomologist that we work with that helps us diagnose those problems and confirm exactly what's going on and give recommendations to the growers. But we have to know enough to be able to identify that it has a nematode, and then maybe the nematologist will help us figure out which nematode it is.
Yeah, like we talked in the first segment, there's a lot of different expertises when it
Yes.
comes to diseases. Bacteria, viruses, and then there's a nematologist, a pathologist. Oh, so many. But let's
Yes,
go to the
definitely.
microphone, to the microscope, because this is a microphone. Let's go to the microscope and just go look at fun stuff, and you can tell us about what we're looking at right now.
Okay, so what I'm going to do is I'm going to isolate from this plant that had the crown rot. So that's what I would do if I don't know what is causing this. These plants are out in our field. They are dying. We have not inoculated them. This is just occurring on its own, and we want to find out what is in the field, what is killing these plants. And the traditional way of doing it is actually to take a little piece of the symptomatic tissue, put it on a petri dish, and see what grows out of it. We want to find out what fungus is actually growing inside of this strawberry plant and killing it, because there's actually a lot of different fungi that could be causing this disease. It's not just one.
And all right, let's get into this microscope and see very closely into the plant.
Let's see. Okay, so I'm going to try to center this up here on the screen so you can see it, what I'm seeing here. Could somebody hand me the scalpel I left over there?
Scalpel. We're looking for the scalpel. Never run with these.
Thank you so much.
This is science happening live, just for you guys.
And I want to make sure that there is nothing on this scalpel, that it is sterile. And to do that, I am going to dip it in some alcohol and flame it to kill anything that would be on the surface there. Okay? Then I am going to make a cut right on the surface here, because I want to get clean tissue underneath here. And So you can see this red, let's see, it moved. So you can see here is the, I gotta flame this. So you can see when I cut into this, you have the red disease, sorry, the scalpel heated up the lens. You can see the red disease tissue here, and you can see this white healthy tissue right here. And so when I cut into this, what I'm cutting away the tissue with a flame scalpel so that I can get to a clean section. And then what I want to do is cut a piece right on the edge of the brown necrotic tissue that's rotten and into the white healthy tissue. So I want to get the pathogen that's moving from the sick tissue into the healthy tissue.
I see.
If I do just the dead brown tissue, I could get, I might not get the pathogen. I might get just fungi that likes to live on dead tissue. We call it saprophytes.
And that would mean that you would get a bad diagnosis, right? If you
Right.
got that bad sample.
Yes. So.
This is pretty exciting.
So I cut in the white and then into the brown. And then I'm going to cut the little piece of tissue out, avoiding the edge of the crown where we might have dirt or things located. So I just want to get right from the center of the tissue here. And
This is very exciting. I really want to know what's in here. What killed this plant?
so then I get that little tissue piece, and then
That is a very, very small piece.
it is a very tiny piece. And I am going to hopefully, so you see it there on the tip, and I'm going to put it on the auger there.
We see it, yeah.
So what we would normally do is we'd have multiple plants from the field, and I would take little tissue pieces like that from multiple plants, from like 4 or 5 plants, and put them all on the same plate, and I'd see what grows out. That way we can see what's killing all the plants across the field, because sometimes it's not just one pathogen. You could get more than one thing killing plants in the same field. And when you can't tell, when all I can see is crown rot, that doesn't tell me what's there. So I'm gonna do pieces from multiple places. Then we have to let this grow for about 5 to 7 days till we get something growing out, and we can identify it then based on what it looks like. It's colony morphology, which is what it looks like growing on the plate. And also we look at the spores that are produced under the microscope to help identify what we're looking at.
Yes, and
It's also, I would also do more than one type of petri dish because different fungi grow on different media. So I can't just do one petri dish with one kind of media. So these petri dishes are food for the fungus. This one is actually made with potatoes and sugar, and then it's got some antibiotics in it to prevent bacteria from growing on it. But it's just potatoes and sugar. We make media from a lot of different things. We also make we make a media from V8 juice. We make media from cornmeal agar. So a lot of times they like to eat some of the same foods we like.
you said V8 juice, like
Yeah.
the one that we
Oh, yeah.
buy in
Yeah, yes.
any grocery store?
Yep.
Oh, we eat the same as plant diseases do, and
Like I said.
as fungi do. That's
Potatoes
pretty.
and sugar in this one.
Oh,
V8
interesting.
juice in a different one. So yeah.
Yeah, so to recap, we're looking at how the scientific process works here. So a farmer has an observation. They see sort of a disease happening in their in their crop or something wrong. They see their plants all wrong, and they send you the sample. You make now an observation. You say, "Oh, this plant, yeah, it doesn't look very good." Like we saw the little diseased plant. Because you can see this doesn't look very good. So, and then you have a hypothesis. You said, oh, I think it could be this. It could be this other pathogen. And then from there you go to your microscope and test your hypothesis. Get your sample and
Yes.
then try to get your
We try to isolate the pathogen and see what we can find. And we had some questions I heard from the audience.
Yeah, let's go to the questions right now and just keep answering this.
Thank you so much. That was so interesting to learn about. We have a lot of questions that are coming in. Our first one is from Steph, and Steph wants to know, so basically a farmer can bring their strawberry plant to you just like a person would bring themselves to a regular doctor's office?
That is exactly true. That's exactly right.
Awesome. And she also would like to know, where do the fungi and bacteria come from?
So it depends on the particular pathogen. So some of the fungi live in the soil and they're already here and they can spread from the soil. So we try to treat the soil ahead of time to prevent that from happening, but sometimes some of it survives, and we call that soil fumigation. Some of the pathogens actually come on the strawberries, and when we get to our weather conditions, like Natalia talked about earlier, we get really conducive weather conditions. So the plants may have looked really healthy up north, and we plant them in the field here, and all of a sudden they start dying because the pathogen was already there. Some of them can actually spread from other plants. So one of the common causes of crown rot, Calochatricum gliosporioides, it can infect a lot of different plants, and a lot of times we'll see it right on the edge of the field, and it might be coming from some of the weeds or oak trees. The fungus can come off of the leaves on the oak trees and can fall on the strawberries and infect the strawberry plants. And so you might see a lot of it right at the edge where there's other plants around. So it can come from a lot of different places.
That's awesome. You talked just a second about treating a field. Can you just explain what that means?
Okay, so before we plant strawberries, we do something called soil fumigation, which is we apply a chemical in the soil that helps kill the pathogens, helps kill weed seeds, and helps kill nematodes, and so that we have to try and give our plants a good start when they start, right? So they don't have so many things that can infect them when they
And
get
are
there,
those
and weeds that compete.
Are those chemicals like dangerous?
They can be. They are chemicals. They can have dangers. They are, but we follow very strict rules and precautions when applying them. You know, they go in the ground. They're covered by plastic. They're maintained, and they have to give adequate time for them to diffuse and inactivate before we plant the strawberries, so they're no longer present when we plant the strawberries.
Okay.
I said they kill weed seeds. They would also kill the strawberry plants if we put it directly in it. So because otherwise we get a lot of weeds competing with the strawberries, and that affects the growth. You don't get near as big of plants, near as many strawberries.
So we won't, at a grocery store, come in contact with those chemicals. They're already diffused, like you said.
Yes.
Okay, awesome.
Yes, we have. You have to give a good month usually before. You usually do it about a month before planting.
Oh, okay, good to know. Thank you. And Steph also wants to know, can people catch any diseases that that these strawberry plants have?
None of the pathogens on strawberries affect people. There are a few fungi that infect some plants that can produce toxins that affect people. But those are all screened for by the public health. The crops that are known to have those issues, they have to do mandatory tox screenings to make sure none of those fungi are growing and that those toxins are not present. Mushrooms are fungi, and if you know, like mushrooms, some mushrooms are healthy. You can eat them. They're delicious and wonderful, but you can't just go pick a mushroom out of your yard, because a lot of mushrooms produce toxins that can make you sick. And that can be true of some fungi that cause plant diseases. Some of them can produce toxins that affect people, but none of them on strawberries do.
That's awesome. Thank you so much for sharing that. And the last question for right now is from Kylie, and she wants to know, how many different strawberry plants does you and your team study at one time?
Oh boy, that is a tough one. So out in the field, we have thousands of plants. So if I just think about one trial I have, and we have probably about 20 different, at least 20 different trials out in the field, but just one trial I have, let's see, I have 50 different treatments, and each treatment consists of 40 plants.
Oh, wow.
We test 40 plants with that treatment, with those treatments.
And how
So,
many trials can you run at one time here?
I'm guessing we have about 20 trials in the field,
Wow.
and we are limited by space because we have to share the field with all the other programs. And sometimes we work in conjunction, but like the breeding program that breeds for that sweetness in the strawberries, and with the entomologist who studies insects on the strawberries, and then nematologist who studies nematodes on the strawberries, studying for people who study like fertilizer, like how much fertilizer to give and what's the best conditions for growing strawberries, and so other things that affect the growth of strawberries. So we have a lot of different research programs, and we have to share the field, but we have a lot of field space too. So.
Awesome. Well, thank you so much. I will be back in just a little bit. I'm going to hand it back over to Cesar. Thank you.
Thanks again, Caitlin. And thank you, Teresa, for answering all of our questions and for letting us know how difficult, lengthy the scientific process and the diagnosis of a strawberry plant is. Thank you very much for being with us.
Yeah, it can be a real issue taking 5 to 7 days. But Marcus, who you're going to talk to next, is going to talk about some of the ways we've addressed those issues.
Thank you very much for that segue. And yes, we're gonna have Marcus talking to us about how new technologies or how technology is actually in speeding that process of diagnosing a disease in a plant. So without further ado, here's Marcus. Marcus, if you want to come to us, and I hear you have some new technology you're developing here in the program and in the lab, and that's all very exciting. We're going to get to that, but first, could you start talking or explaining to us what new problems strawberries are facing, what new diseases or just problems in general?
Oh, sure, no problem. So like Natalia was mentioning on the first segment, here in Florida, because the weather it's warm and humid, so we have a lot of issues with diseases. So here in this banner here, for example, we have an example of different pathogens that can affect any strawberry and the different diseases that they can cause. But recently, we had a new disease that emerged on the strawberry that is caused by a fungus called Neopistalothiopsis, and this disease started like in a few fields and quickly spread it through the strawberry industry to other fields, other farms, and other states, and it's like COVID, So it's a new disease that happened. The science still need time to figure out what to control, what to do, from where it came from, but we are working on it. So this is an example, for example, like a strawberry plant that is infected with this disease. So you see all the necrosis on the leaves. It is pretty bad.
The necrosis is the dead tissue, the brown
dead tissue
part of the leaf?
So what happened is like all that necrosis, it limits the plant to do photosynthesis or to produce food for itself. So eventually, because the plant cannot produce food for itself, the plant starts to get stunted and eventually die. So here, for example, we have a picture of a field that was highly infested, so you can see like 100% of the plants they were infected. Okay.
And that new disease and the old diseases affect how much food and how much strawberries there are in a grocery store. So we gotta say thank you to the scientists who are thinking about these new methods and trying to find out these diseases so we can keep having strawberries with us.
Yeah, and then what's happening is like we have this new disease and also the old disease going on, but like Teresa was showing us on the previous segment, we have to take the disease, the symptomatic tissue, put on a culture media that is basically a petri dish with food for the pathogen to grow, but that takes time. Like 5 to 7 days. It's not like the disease is waiting for us to give results for the growers. So we need to have a quick answer. So that's when our new methods and new things or new tools that we are working on to fast that process. Okay. So one tool that I can show it to you right now. Maybe if I can have Andrea here, and I believe I do have a slide with that shows a workflow for the DNA. For the DNA part. So for example, we got a symptomatic crown that is showing the crown rot symptoms. So we get a piece of that plant and we extract DNA. So that DNA, here we have Andrea, our lab technician, and what she does, she have a special plate where on each well she can load a different sample. Okay? So she load that DNA that came from the different plants, and then we have this special machine here in our lab that was donated by the Florida Strawberry Growers Association because they really recognize our work and everything we do. So that plate goes inside this machine. So what this machine will do is gonna read that DNA that we are putting on the wells of the plate, and what happened is like each pathogen have a different DNA composition, and that different DNA composition can be captured by this machine, and they have different curves, like a different fingerprints that every person has one, and those fingerprints of the DNA is gonna tell us which pathogen we have in the sample. Okay. So for example, here we put the plate, and then we have control DNAs that will be our positive sample. So we have control DNAs for different pathogens, and then what do we do here in the machine? We selected, for example, this sample here. Whenever it's blue, it means negative, no reaction, and whenever it's green, it means it reacted with something. Okay, so if we see here, it reacted with the pathogen that has this blue curve. Okay, so among the 4 other pathogens we are testing, we can see that our sample is lining up with the blue curve. And that blue curve is Phytophthora. Okay, so based on this method, once we receive the sample, we can tell the grower within 24 hours a result and let them know what they have in their field, so they can start treating and hopefully do not have much implication on the yield.
So with this DNA seems to work very fast and to be a lot faster than traditional methods for diagnosis, which in turns help the growers knowing what their diseases are. Have what diseases they have in the field, and they can act very quickly. Mm-hmm. Can you tell us a little more about what are the benefits of knowing that quickly about what disease you have in your plant and in your field?
Yes, sure. So for example, the Phytophthora issue, that was the case for this sample. If we know that the pathogen is already happening in the field and some plants are already getting sick, if you treat the plants in a timely manner, you might be able to recover some of those plants. Okay, so that process is really important. Or in the case of Neopistalothiopsis, for example, you have a limitation on what you can do, but if you know that you have a positive plant or in a certain area of the field, maybe the grower can go there, remove that plant, or they know that they can treat their area for last, so they don't cross-contaminate other fields. So there are a lot of implications that knowing a result fast can help them out.
And that way they also save money because they don't have to spray everything or do that much work, so they know. I also heard that there's something even faster that you came up with or that you have in your lab. Could you tell us a little more about that? And we can walk and you can show it to us too.
Oh, sure, no problem. So actually, yes. So this one, for example, like that sample was positive for Phytophthora, okay? So there is a commercial kit from Agdia, and we call it Immunostrip because this is like the fast COVID test, so like we do at home. So you get a sample and then you put in a solution that came already from the company and you insert a strip. And if you have, for example, 2 lines, that's what you have here on this sample, it means it's positive. The line on the top is the control. That means, okay, my test is valid and my test worked. And the line in the bottom, it means if my sample is positive or negative for the pathogen, okay? So tests like this are already available for some of the pathogens that growers or people or nurseries could do actually out in the field.
That's amazing. So yeah, they can save a lot of money, and I see that you guys are getting new technologies into the hands of the growers, which is incredible. And well, maybe we'll talk a little more about other things, but right now let's go back to the questions and answers. Let's see what the kids are thinking and how you can help them figure that out.
No problem.
Hi again, I'm back. We have a lot of great questions coming in. Alicia would like to know how long it takes to develop the method with this machine.
Hi Alicia, thank you for watching and thank you for your question. So it varies from pathogen to pathogen, but I would say like almost a year to get the test up running, okay? Because you have to first study the pathogen, know the DNA, figure out how you're going to develop the test, and of course you have to do validation of the test to see if your test is accurate and is already can be employed in the clinic. So there is a lot of like studies to be done in order to be confident that the test work and we can put this available to growers.
Awesome.
Okay.
And Alicia also wants to know how many diseases can you identify with this method?
Yeah, so with this method right now we can identify 5 crown rot diseases and the Pistalucia leaf spot, which is the new disease that's going on the strawberry that I was mentioning earlier.
Okay, awesome. And what disease is most prominent in Florida?
So it depends on the season. It seems like every season, every strawberry season, which start in October and end in April, March, it depends, So, for example, right now we have a lot of Coledotricum acutatum that's causing anthracnose and root necrosis. We have some Pistaluciopsis going on. But yeah, every season have its challenges, okay?
Gotcha. Awesome. And I think that, well, Stephanie said this is awesome, and I agree. And she would like to know what advice you have for students who might want to be plant pathologists.
Hi, thank you, Stefan. Yeah, the advice I have is like you really have to love what you do. And I think if you put love and if you find yourself in a position that you really like, especially if you like to help people figure out why the plants are getting sick, how to save them, is like I think it's a very noble path to follow, because disease is going on all the time, like happen with human beings, and it's very awesome to figure out what the disease are and also what to treat them, so you can save the whole community and keep strawberry healthy.
That's super awesome that you get to literally help so many people all the time with your job. And that's all the questions I think that we have been asked today. But we have been doing polls throughout, and one poll that we're doing right now is, do you like strawberries? I'd like to know.
Oh yeah, I love strawberries. They're very tasty, you know. I love them yeah. That's the best part of my job, you know, getting to taste those strawberries out of the field and out of our trials, you know.
Okay, awesome. Thank you so much. We'll grab this back to you.
Thank you, Caitlin. And thank you, Marcus, for sharing all of us the new technologies and how they're making a grower's life better. And thank you for being here and answering your questions.
No, thank you. Thank you everyone for watching, and for you guys that organized this. We are very, very happy.
Yeah, now that we know what the Plant Diagnostics Lab does, let's now we're gonna go to the field. So don't forget to tune in for November 16 to know everything and learn everything about what they do in the field, so they can have strawberry plants to study. And that's it for today, everyone. But before we go, let's thank our scientists and say bye to everyone. Bye, Dr. Perez, Teresa, Marcus,
Bye.
and Andrea back there. Thank you so much for spending time with us today, and thank you to the Strawberry Science, to Streaming Strawberry Science team for making sure that we all get this at home. And just thank you so much, everyone, for make science a little sweeter, and for making the strawberries a little sweeter too.